Hi, everyone. So my protein that I had was tetrameric phenylalanine hydroxylase. And for some basic information on how this macromolecule functions, essentially in mammals, it catalyzes the rate-limiting step in phenylalanine catabolism. And what that means is it actually helps convert L-phenylalanine to L-tyrosine through hydroxylation of the aromatic ring. We'll get into that mechanism a little more later, but it essentially requires the cofactor tetrahydrobiopterin, or BH4, to be present in order to carry out the reaction. And so ultimately, the synthesis of L-tyrosine is further degraded into products that aid in different actions in the human body, such as the citric acid cycle, as well as the creation of neurotransmitters. And you can see here that eventually some of the products are used up in the citric acid cycle. And so PAH is actually mostly found in the liver, where it removes excess amounts or toxic amounts of L-phenylalanine. And I think something that's interesting is that in bacteria, one of the interesting functions is that it actually aids in the synthesis of melanin-like pigments. And this can kind of explain why people with phenylketonuria, or PKU, have pale hair color and pale skin color. And this is the mechanism in which tyrosine is created into synthesized or used to synthesize melanin. And there's a lot of information on the structure of the molecule, but I kind of highlighted the most important information on this slide. So basically, 4 PAH molecules interact to form the tetramer, which is the functional unit. And each molecule in the tetramer is organized into 3 domains: a regulatory domain with an N-terminus residues 1 to 110, a catalytic domain where all the enzyme or most of the enzyme activity is going to occur, which is residues 111 to 410. And that's also where the BH4 is stabilized by the iron atom at the core. And then finally, the tetramerization domain that assembles the 4 chains together, which is the C-terminus residues 411 to 452. And so this is just detailing more of those interactions between the amino acids and the substrate, and as well as the iron and the BH4. So this is the actual output I got from the PDB code I used, and it's an X-ray diffraction image. But some of the most important information to take away is that it's composed of 2 unique chains, an A and a B chain with length 335, and it's of course taken from Homo sapiens. And the total molecular weight is 77.40 kilodaltons, including non-water atoms, basically, in the asymmetric unit. And then the atom count is 5,317, not including hydrogen atoms. And then the residue count is 651. So for some basics of the molecular interactions, like I said before, it requires the presence of BH4, and that is stabilized by the iron. And you could see the complex sort of right here in the image. And during the course of that reaction, the cofactor loses its 2 of its hydrogen atoms to form the di form, dihydrobiopterin. And then another enzyme ultimately reacts with this to restore it to its original form, where it could be used for another cycle of hydroxylation. And of course, mutations can block the conversion of phenylalanine to tyrosine. And there's many different mutations, but some of them are shown in red in this structure. And of course, this is what's going to lead to diseases such as PKU, which we briefly discussed before. And of course, there are also other serious mutations that are not shown. So for mechanistic considerations, which is really important, phenylalanine is obviously an essential amino acid, L-phenylalanine. And so after it's ingested, it's integrated into various proteins, oxidized to tyrosine, and then converted to other metabolites. And that occurs by the hydroxylation system and the enzyme PAH. And you can see here the 3 domains, the catalytic being the heart of where all the action takes place. And also, it functions as the rate-limiting step for the oxidation of phenylalanine to additional products of CO2 and H2O, which ultimately contributes to the formation of glucose and other 2-carbon metabolites. And this is obviously the main mechanism, where you could see the coordination of the hydroxyl group on the tyrosine is in the para position. And also further mechanisms state that after a protein meal, when you ingest the phenylalanine, it causes a release of glucagon from the pancreas. And there's a ton of other steps that happen, but the important thing to take away is that this is regulated by protein kinases and sort of a cascade, a phosphorylation cascade involving the secondary messenger cyclic AMP. And this is ultimately going to influence BH4 binding with PAH and will enhance the activity. And in terms of enzyme kinetics and regulation, the studies have sort of revealed that it's allosterically regulated by the substrate, the cofactor, and phosphorylation, and that BH4 is necessary in order for the substrate to bind more efficiently to PAH, and it raises the activation. And uniquely, you could see in the curve here that it's not your typical hyperbolic Michaelis-Menten enzyme kinetic curve, but rather a sigmoidal curve. You can see the S shape sort of forming at the bottom and then the top. And that is the Hill kinetic model. And then also, like we stated before, BH4 is required. And the reason why is because it is positive, it has positive cooperativity with PAH. And you can see that with a Hill coefficient greater than 1, which is 2.2. And ultimately, when it's not bound to BH4, PAH is going to have a Hill coefficient of only 1, indicating that it's not cooperative. We also see the Km decrease from 24 to 8 micromolars in the presence of BH4, which indicates that L-phenylalanine has a greater affinity for PAH with BH4. And you could also see, because of missense mutations, which lead to PKU and PAH not functioning properly, on the bottom mass spec image here, you have phenylalanine not being converted to L-tyrosine, and you can see it's not detected by the mass spec like it should be in the top image. So this ultimately leads to the implication in human disease, where a mutation in the PAH gene in both of the alleles on chromosome 12 causes PKU. And this is passed down in an autosomal recessive manner, meaning that the child can only develop it if both parents pass on a mutated version of their gene to that child. Of course, parents could also be carriers, but in order for the kid to even have a chance of having PKU, both have to be carriers. And the occurrence is very small in the US, but combined with other inborn errors of metabolism, the occurrence of these diseases is actually quite high and affects a wide group of people. And so in those with PKU, PAH activity is obviously reduced, and there's less affinity for L-phenylalanine. And that leads to a buildup of toxic concentrations of that amino acid. And some of the effects are irritability, seizures, dry, scaly skin or eczema, musty or mouse-like body odor, and pale hair and skin, and developmental delays. And you could see the white brain lesions actually in people who have unfortunately not detected the fact that they had PKU at an early age. You could see the toxic buildup, what it does to the brain. And so the reason why brain development is so affected is because of the fact that L-tyrosine, which is the product of that reaction, is used for a lot of different metabolites that cross the brain barrier, as well as a lot of different neurotransmitters. And so because of this, less tyrosine is synthesized, and tyrosine is actually a precursor to dopamine. It's converted to L-dopa. And so if you have less L-dopa, you're going to produce less dopamine. And because dopamine is important for mood, learning, memory, and motivation, this explains the developmental delays and lack of cognitive functioning in those with PKU. And so if it's left untreated, ultimately it's going to cause that irreversible brain damage and cognitive decline, and could lead to more behavioral, emotional, and social problems later on in life. And this is the mechanism for the reaction of tyrosinase and tyrosine to form L-dopa. But luckily now individuals can avoid phenylalanine in their diets, and also take BH4 supplements to sort of enhance the ability of PAH to function properly. Thank you. That was my presentation.